All right, we'll wait just a few more minutes and see if we have any more attendees, and then we'll get started. Could be a lot of people are planning on watching the recorded version of this based on the registrations we have. All right, I think we can get going. Um, so welcome. My name is Charles Simon. I'm the program director for the Blueprint Neuro Therapeutics Network. Thank you for taking the time to listen and learn about the Blueprint Neuro Therapeutics, otherwise known as BPN. Um, I'm going to talk to you today about the program, how it operates, and then give you some other programs that are out there that can give you the data you need before, as well as some complementary programs for biologics and pain therapeutics, and finish on um, some things to do and some things not to do as you prepare your application. So I want to make sure we are not paused anymore. Perfect. So the program's vision is to combine the strengths of, and you're probably seeing this up here, which you don't need to see. Sorry about that. Is the program vision is to combine the strengths of NIH and industry expertise in order to move your projects forward. We look for you to bring your investigator-initiated ideas for novel drug targets and disease assays and models, along with a starting point, chemical starting point, and we can provide experts in terms of advisors with extensive pharma experience, as well as access to industry standard contract services. That's up here, make sure, there we go. So the overall goal here is to de-risk the potential therapeutics to the point where industry will invest in them and allowing the projects to continue efficiently towards patients. We're really looking to identify the best ideas for translation in the community and provide the funding opportunities and the associated infrastructure to move them forward. This funding is non-dilutive, where we set the contracts up in such a way that we're trying to keep your intellectual property with you and unencumbered in order for you to license it at the end in the most efficient way possible. Um, the program is for academics and small businesses. It is, the Blueprint Neurotherapeutics Network is targeting small molecule therapeutics, but there is a complementary program for biologics, and I'll talk about that at the end. Um, the the grants support projects from discovery all the way through the development phase, and you can enter in anywhere along that paradigm, and we will give you access to not only funding for the science and disease biology efforts in your labs, but also access to contracts if you don't have them already established for consultants for CRO access, as well as access to NIH expertise in terms of project managers and science officers that really know the landscape and the portfolio around your disease area. The way the infrastructure is organized, we create a lead development team for each project, and that consists of you as a principal investigator, along with industry consultants that we'll assign to you and NIH staff. We set it up very much like a pharma model. So we meet every 2 weeks and we discuss the activities everyone's doing and prioritize next steps. As I said, the grant can certainly provide and requires that you have the disease biology expertise, the SAR driving assays for in vitro and in vivo for your disease indication. If you don't have the medchem synthesis expertise or the in vitro ADME or uh, in vivo PK or toxicology or any of these other things like drug manufacturing formulation, clinical trials expertise, we can help that out through our contracts. If it's in a discovery phase, everyone uses our relational database system to put their data into. And it's collaborative drug discovery does that. So you really have to ask yourself, do you have the expertise and resources in-house to do these activities, or do you need us to help you? And if you do, talk to us about it and we can help um, you figure out exactly how to, what we can and can't do for you. There are 9 participating ICs that are signed up to take projects under the Blueprint Neurotherapeutics Network. And if your project fits under one of these ICs, institutes or centers, then we can take your project. And these are the science officer contacts at each of the ICs that you can talk to about your program, make sure that you're within the mission, and then contact me. And I can walk you through the process of how to apply and how to structure uh, your application for the best results. As I mentioned, you're coming in anywhere along the pathway, but you can also exit anywhere along the pathway. And coming in early stages, some people have exited at the phase 1 stage, some people have exited at the IND enabling stage, um, and some people have exited at the lead optimization phase. But in all cases, from a grant perspective, you're right in the grant as if you're going to phase 1. And you may license it earlier and you may take it out of the BPN, or you may license it and stay within the BPN and continue to work, and they may take it right after we're done in phase 1. But the grant application is certainly set up to go all the way to phase 1 in pretty much every case. It is a 2-phase mechanism. We have both a SBIR version and a non-SBIR version of it. The U44 is the SBIR, and the UG3, UH3 is the non-SBIR versions. So what does that really mean? It means from a grant perspective, if you're an SBIR eligible organization, that you should probably consider the SBIR funding mechanism because it does offer you some advantages. But if you're a small business that is not qualified for SBIR, you can still come into the UH3, UG3 mechanism as well. The preparatory phase, everyone enters in that first phase of the grant. It's really about establishing your team, identifying, conducting the necessary studies to meet the BPN requirements for the stage that you're coming in at, design and executing plans for a no-go decision or go-no-go decision at the end of that first year that will determine whether you move to the next phase, and then progression through each phase will be based on an administrative review as well as following milestones throughout the project. So in the first year and every year subsequent, we look at milestone progression. In the UH3 phase, it's BPN typically supports up to 2 years of medicinal chemistry in this phase and 1 year here, so a total of 3. If you were to start at the early discovery phase, and if you're in later stages, you may only be 2 years or 1 year, depending on what you're doing. So it really does depend a bit on where you come in. And we do tell you it's important to not overestimate where you come in, because you'll see in a moment about the milestones. It's important that you fully meet the milestones in order to move forward to the next stage. We do support IND enabling studies and first-in-man trials under this program, and we mainly do those through contracts in most cases, but there have been occasions people have proposed to do themselves. When they do, they have to obviously We'll talk about this in a moment on my next slide. Whoops. All projects have to fall under one of these two things. But if you're under the UG3, UH3, and you go over 500K in direct costs in any year, in UG3 or the UH3 phase, you must apply for ARA process at least 6 to 8 weeks in advance. So it is important if you're coming in under this mechanism that if you're going to be over 500,000 in direct costs in any year, you think about doing everything really early in the process so we can get you approved to come in that way. It is also important to note that if you're coming in at a development stage project, not all ICs accept development projects. So for instance, NIMH and NIDA are not that interested in doing development projects under BPN. They have their own mechanisms for following up those kind of projects they prefer you to utilize. If you're cutting on a U44 program, again, these are the dollar amounts that are listed for NINDS. But if you were to come in under NIDAs, their rules are slightly different. So again, be careful on what you're thinking about and which disease area, because each IC has its own set of rules. And we want to make sure you again, talk to us early so we can tell you what those rules are. The next application due date is in February, on February 9th. And again, applications from foreign sources, I mean, we do accept foreign applications as well as foreign components, but if you're a foreign application, meaning the main institution is a foreign institution, there's a second level of review that goes on, and reviewers will assess whether the project presents special opportunities for furthering research programs through the through the use of unusual talent, resources, populations, or environmental conditions that exist in other countries and are either not readily available in the United States or augment existing US resources. So you have to make that sort of extra case for it moving forward. As I mentioned, we work on a milestone progression. And so a lot of people do get confused when they look at specific aims and milestones. You have aims that you lay things out in, and it really should sort of follow this path of a standard drug discovery paradigm in small molecule space. And it should take you all the way from wherever you're entering in to the clinic. Even if you're not running the clinical trials, you should say at the end of this, we're going to utilize NIH CROs to do these things so that the reviewers understand that you're going from all the way from here all the way to the clinic as part of the program, and you lay that out. It is, essential that you see this progression, and this is how we do progression from one phase to another. So for lead optimization, for instance, we're looking that you have at the end of lead optimization, your efficacy done, and so you have a PK, PD relationship, as well as a rodent dose range finding tox study so that you can do therapeutic index. These can be run through our CROs. This is going to have to be part of your grant. Sorry about that. The efficacy studies. As we move later in this program, these are almost exclusively done in CRO work, either through your contracts or through ours, in order to get the project through the development phase, because you're basically checking boxes for the FDA at this point, and everything has to be done according to regulated procedures. Intellectual property, I touched on at the beginning. Inventorship is always determined by U.S. patent law. The blueprint itself is not seeking a stake in your IP, and we set up our models so that we avoid NIH staff people becoming inventors on your program. The extramural staff are here to facilitate. We're program managers. We keep things operating, but we don't get involved in telling you exactly what molecules to make and so forth. But prior to the grant award, we will ask that you have IP agreements in place with all potential inventors from our contracts, from our consultant contract group. So if we assign you consultants, we'll ask you to have intellectual property agreements in place with those consultants to say they're going to assign the IP to your organization and you have it worked out. The NIH staff don't sign those. We can't. But again, we're not trying to become inventors on your projects. Our MedChem and drug manufacturing formulation contracts have clauses in them. They're called determination of exceptional circumstance clauses that they, by contract, are required to assign the intellectual property to you. And the overall goal and for yourself as well, if you have people you're working with, is to have unencumbered IP and to make sure that all the agreements are in place. So if you're working with multiple institutions, we'll again ask that you have a letter that says that you've already agreed who's going to handle the intellectual property and how it's going to be assigned. We have a great web page that has lots of information on it, and this is the link up here at the top. And on the side, you can see, what our current development projects are, what the funding opportunities are, who's on our external oversight committee, and who our consultants are. Has a frequently asked questions area. It's a great resource to certainly look at and find more information about the program, may be of interest to you. But what if you don't have all the entry criteria? If you don't have good, preliminary data for efficacy that you may want to see, or you don't have the chemical matter quite fleshed out the way you'd like to have it fleshed out prior to entering the BPN. There's a program, at least for the NINDS mission projects, called Ignite that can help you get that data. There's a project, a PAR announcement for developing an assay and running a screen. There's one for, doing preliminary SAR and running an efficacy study. There's one for developing an animal model and then testing that animal model out. So these are great resources for getting preliminary data if you find that you're not quite ready for BPN and you need to develop those additional pieces. They have receipt dates coming out in February and June. It works for both small molecules and biologics, and Rebecca Roof is the contact for it. It's also a phase mechanism, so you'll come in, but it's not a cooperative agreement, so you're not going to be meeting with them every 2 weeks like you do in BPN, but you will have to set up a go no go milestone as part of the application, something that warrants going from the first phase to the second phase. But you can have a total of 3 years. It could be a 2-year or 1 year first phase and a complementary number of years for the second phase. But you can only have one no-go decision point in there, and you have to have a clear milestone laid out as part of the application. So let me cover some things to do here, and then I'll come back to some of the other programs. Read the opportunity carefully. There are things requested in the FOAs that often are missed by applicants. Things like it requests tables of who's going to do, what's the assays are, what the throughput of those assays are, who's going to do them. It asks for intellectual property summary in one page as a special attachment under the UH3 mechanism, whereas in the SBIR is looking for a commercialization plan that would be part of that. You have to stick to the page limit. You can't go over the requisite number of pages, and you can't stuff by using microfont because that just upsets the review staff and can end up with your application being withdrawn. Do put forth a solid scientific premise based on your preliminary data and address the rigor of that data in your research strategy section. A lot of people don't address the preliminary data and how rigorous that was, so it is important to do that. And it's also important, I'll talk in a moment about rigor in all your data. Address the obvious criticisms of the chemistry and biology. I suggest you contact the missile chemist or BPN staff if you have questions about whether you have a structural alert in your molecule or some other issue that you may want to at least think about addressing. And clearly indicate what will be done as part of the grant and what you expect to be done by BPN contractors. That way we can tell you up front, and if you talk to us in advance, we can avoid any confusion because BPN contractors can't do everything, and we can't do a lot of one-off things. It's meant to be, used as part of a program. Do include budgets for all years of the grant. So not just the first phase, but for the entire 5 years, if it's a 5-year grant, give a budget for every year of the grant. As I mentioned about rigor, it's important to discuss the preliminary data and supporting data, but it's also equally important to talk about how your data will be generated in a rigorous fashion as part of this grant. The definitive efficacy studies, when you go to run them, they need to be blinded and randomized and have exclusion and inclusion criteria and proper appropriate power analysis. You need to highlight all of this and not hide it in the vertebrate animal section, but have it as part of the research strategy section on how this data is going to be done in a rigorous fashion. Do complete your registration 6 to 8 weeks in advance. Every cycle I run into someone who's calling me at the last minute saying, I haven't got my registration yet. And it certainly can take a while for that to happen. So I recommend at least 6 to 8 weeks in advance. Likewise, the same thing if you're going to apply for greater than 500K in direct costs in any year of the grant, I still need that 6 to 8 week submission window in order to get you the approval to come in. You need to submit me a very detailed budget at that point, and we can discuss with you what needs to be done. So if you're going to do that, contact me early. So consider submitting your application early. Again, every cycle, I see applications end up getting withdrawn because they haven't followed something in the rules. And if you send it to me early, I have an opportunity to look at it before the deadline, and and perhaps it'll give you an opportunity to correct the problem and not have to wait another cycle to come back in. Do talk to your tech transfer folks and work out those IP agreements as well as plans for patent filings and so forth down the road. Some things not to do: don't plan on this to be your sole source of funding for your lab because it's milestone driven. It can end abruptly if we don't meet milestones. Um, do not under-resource the budget to avoid the limits I talked about. Don't put 499,999 down when in fact you need 750 in order to do the work, because it's not easy to deal with the getting a supplement later. It would be better to have it in under review to do the ARA to get the permission for going over the limit up front. That way review can comment on the need for that budget, and it makes support of the larger budget a lot easier. Do not plan for the BPN contracts to do disease biology, and as I mentioned, or one-off sort of experiment. So if you need to run an AIM study, and that's the only thing you need, don't look for the BPN contractor to do that. You should just contract that yourself. It's a lot simpler and easier. It's a lot of work to do government contracting, and to do it for a single experiment, it's not cost effective. Do not code your application as being a clinical trial application. If you plan, or human subject application, if the human subjects in clinical trial are going to be done through BPN contracts. That means those contracts have to be labeled that way, but not the grant. The only way you label a grant as a clinical trial grant is if you are proposing to do a clinical trial through the funds that are part of the grant. In which case, in most cases, it'll be delayed onset. We are accepting new applications for both these things on February 9th. Every time people ask me how it's reviewed through a special emphasis panel. And we have the following indications areas for the different ICs, and in the back of the funding opportunity announcements, you will see what their interest is listed as well as contacts for each of the institutes. As promised, I'd also mention and touch on a couple of other programs, just in case you're on here and you're thinking about, oh, I have a biologics program. There is now a BPN biologics program. The contact for that is Mario Skiodopoulos. And it's designed for peptides, proteins, oligonucleotides, gene therapies, cell therapies, and other emerging biotechnologies. It's set up very similarly, and we're in the process of leveraging some of the contracts we have under the small molecules and establishing some new contracts for manufacturing formulation for biologics. Um, those should be ready in the springtime, long before these applications would be funded. Um, Same thing, they have a phase mechanism. They have a SBIR and non-SBIR. The biggest difference, I think, in this biologics is they have up to a 2-year UG3 phase, which we don't have in small molecules. Same receipt date as well. There is a question and answer webinar coming up for that group on December 20th from 1:30 to 2:30, and this is the Zoom link for that, if in fact you need it. And I'll see if I can get that pasted into the chat later. If you happen to be working in the pain therapeutic area, BPN does take pain, but it doesn't take pain for every pain indication. So things like strictly diabetic neuropathy or chemotherapy-induced pain, which fall into ICs that are outside of Blueprint Neurotherapeutics, could come under this, as well as any of the other ICs that are in Blueprint Neurotherapeutics. Again, it's designed very much like Blueprint. It has some subtle differences in that it's seeking to have a companion target engagement biomarker as part of the program, if at all possible. But otherwise, it's a very similar program. Its next receipt date is February 15th. And if you're interested, you can contact me or Marianne Palamounter for that. So with that, I'm going to end and I'm going to start taking questions. I'm going to just go forward here. The application, I can talk about application process, the review process, proposals and budgets, contracts, intellectual properties, any of that stuff will be fine to field questions on. And in the meantime, then I'm going to see if I can get that other thing posted that I mentioned. So I can start to take questions. Let me see if I can escape this.
Hi, everyone. I did post in the chat that there is a question and answer feature at the bottom. We don't have any right now, but we can definitely take the time to get some questions and answers. As well as, I would like to mention that this presentation has been recorded and will be placed on our YouTube page in the next couple of days, and I will be sending the link along with the presentation slides to everyone that has registered. So we'll wait a couple of minutes to see if anyone has questions.
And I did paste that link into the chat for people for the BPM biologics as well. Dun, dun.
Chuck, we have one question. It may be easier if I just read them, if that's okay.
That's fine.
Hi, Chuck. Thank you for this helpful presentation. Does STTR work for this or only SBIR?
SBIR. There's not an STTR version of it, but that's not to say a collaboration between a company and an academic institution couldn't come under the UG3 UH3 mechanism where it could, but there's no STTR set aside FOA for this.
Okay, next one. Thanks, Chuck. Really informative. I think my company is a good fit for this. We submitted a NINDS phase 1 SBIR grant with a proposed start date in April. I'm not sure if that'll be funded when I submit the BPN grant in February.
That's okay. As long as you have the summary statement for the first application, you can apply again. You can't have two applications under review at the same time. So if you've gotten your summary statement back, you can go ahead and apply for the BPN. If it turns out that you're funded under the phase 1, you can always withdraw the Blueprint application at that point. If in fact you're not funded, then you're ahead of the game.
Okay. So we have so far.
Okay. We didn't have a ton of participants today online, so we may not have a large number of questions today. It's unusually low, but maybe with the new COVID world here, we got more people listening to the recorded version than we are getting this. I will tell you this one, that Recondo manages my calendar. So when you reach out and you want to talk to me, I'm going to refer you to Recondo too to set up the time to do so. So feel free to just reach out to both of us at the same time if you like. Just like Elena actually gave me a revised link for the biologics webinar. So the one I posted must have been older. So we'll go with the new one to keep that going. Sounds like there was trouble getting into WebEx. That's not a good thing today. May explain some of the issues. I know we're having a lot of problems today with WebEx, unfortunately, as well as Zoom. And I don't know whether there's a connectivity issue going on right now to get into the NIH world, but it seems to be some stuff going on today.
Another
Well,
question.
if there are no questions, like I said, feel free to reach out to me via email. I'm more than happy to answer those questions.
We have one more, Chuck. Is there a set number of applications to be funded under UG3 mechanism?
No, there's no set number. We look off, they're not percentiled, so they're scored applications. We look at any application we believe to be in the fundable range and do our due diligence on that
one. Okay. I think that's all.
All right. Well, thank you very much for your attention today. I really do appreciate your time. Enjoy the holidays and wishing all health and a prosperous new year. Thanks now.